Hello, I'm Dr. Eric Daar. I'm a professor of medicine at the David Geffen School of Medicine at UCLA. And today I'd like to discuss recent data on investigational anti-SARS-CoV-2 monoclonal antibodies in development for prevention and treatment. As you're aware, monoclonal antibodies were an extremely important part of our therapeutic and preventative strategies during the early years of the COVID-19 pandemic. And they've had a big role in both treatment and post-exposure prophylaxis and pre-exposure prophylaxis. However, over time, with the emergence of new variants, they have increasingly lost their activity against the predominant variants circulating in the community and have no longer been authorized or approved for prevention or treatment in many months. This continues to be an important unmet need, particularly in the area of prevention, because we do have other therapeutics, particularly our oral agents like nirmatrelvir, ritonavir, molnupiravir, and our intravenous remdesivir, all of which continue to be active against the new and emerging variants. But for prevention, especially for those 3% or so of those in the United States that are immunosuppressed and may not respond well to vaccines, we have no currently available strategies. So monoclonal antibodies continue to be looked at. It's also important for us to be thinking about these emerging variants because we know these have been the Achilles heel of monoclonal antibodies. And the figure demonstrates what's been provided for us for some time based on sequencing data in the community. And you may recall in the past, what would happen is we would have a predominant 70, 80+% of variants being of one type, and then suddenly a new variant would come along and take over. Well, we've not seen that in a while. Instead, what we've seen is this convergence or co-circulation of different variants over time. And you can see right now, we still are primarily dealing with these Omicron subvariants, and the predominant ones are the different XBB and EG5. And this continues to be what we're mostly looking at when we think about the development of new monoclonal antibodies and susceptibility to circulating strains. So as far as monoclonals in development, there's two primarily at this time. There's VYD222, which is in very early stages of phase 1 development, looking at escalating doses for safety and tolerability. And then there's AZD5156 product, which is a combination of long-acting monoclonal antibodies. One of them is familiar to you, it's cilgavimab. It's being combined with a new novel monoclonal antibody called AZD3152, which also has an altered FC portion, which prolongs the half-life and FC effector function and complement binding, just as it did for Tixil, now for this combination in 5156. And the key is looking at its susceptibility and then introducing it into clinical trials. And previous discussions with the FDA have suggested that they will weigh heavily the data that comes out of neutralizing titers in vitro and thinking about the development and potential authorization and or approval of future products. So what do we know about these new monoclonals, and particularly this AZD3152, which is the new agent? So this is some data. It's commonly done in these types of antibodies to demonstrate, since they target receptor binding domain, to see whether it blocks the binding of the receptor binding domain to the ACE2 receptor. And they often will use a control antibody, CR3022, which is an anti-spike that does not compete with ACE2, to see whether the new monoclonal is more effective at inhibiting this interaction. And in the figure, it clearly demonstrates that there's a dramatic reduction in binding seen with the new AZD3152 compared to the control. In looking at the activity of this novel 3152 monoclonal antibody against a host of variants, starting on the far left with the ancestral variant, and then those that are familiar to you, alpha, beta, delta, and so on. And then as we move along, you can see it's tested against other variants that had been predominant in the past, like BA.4/5, and then more recently, the XBB variants that we're now seeing frequently in circulation. And if you look at the neutralization IC50, you can see for most of these variants, there is susceptibility. However, when they look at these variants, when there's the presence or absence of 456L mutation, it has a pretty dramatic effect on some of them with regards to their susceptibility. And you can see in pink that if you add to the XBB151 or EG5, this 456 mutation, you go from highly susceptible to highly resistant with a dramatic increase in IC50. And if you look at the table, it further demonstrates what an impact 456 has. Looking at the ancestral strain 614G, there's a very modest reduction in susceptibility. But for BA2, BA5, and importantly XBB 1.5, it goes from very susceptible to extremely resistant with the introduction of this single mutation. And this mutation is not associated with increased virulence or escape from immunity from vaccination, but does appear to be associated with increased transmittability. In fact, recently, it was identified of a variant of interest in the EU, where they've seen a dramatic increase in the prevalence of these Omicron subvariants with this 456 mutation, which could be problematic for new monoclonal antibodies in development. This is additional animal data focusing on the activity of 3152, looking at viral suppression in lungs at day 3 and 5 from a hamster, or 3 and 7 from a hamster model. And you can see in the figure at day 3 and 7, it's most obvious at day 7, that there's a dose response as you go from a dose of 0.67 milligrams to 2 to 6 milligrams compared to the control. You see a pretty dramatic reduction in lung levels. So finally, obviously the important test in thinking about moving a new monoclonal antibody along that looks promising based on in vitro susceptibility is to determine if it actually works. And the Supernova trial has been started. It's a phase 1 to 3 study to assess the prevention of symptomatic COVID-19 in immunocompromised patients at least 12 years of age. It'll include a safety cohort in phase 1, phase 2, a sub-study to evaluate safety, efficacy, PK, and neutralizing activity of AZD3152, and separately evaluate the safety and PK of the combination of the AZD5156. And the primary endpoint, depending on the study and sub-study, will be safety of the combination monoclonal antibodies as well as neutralization activity. And the first participant was dosed on December 2022. So we're looking forward to seeing hopefully new data on a promising monoclonal antibody strategy for the future. Thank you so much for joining me today, and please subscribe to Exchange CME's YouTube channel and check back regularly for updates on COVID-19.